This will be part 3 for practice guided problems in genetics and 2 trait crosses. In pea plants, the green color is dominant over yellow color. Let's take a note on about the colors. So capital G will be for us the green pea color, and lowercase g will be the yellow. As well as the tall is the dominant allele in plant heights, capital T will be tall, and lowercase t will be short plant. Parents heterozygous for both traits are crossed. So parent 1 is heterozygous for both of them, capital G, lowercase g, capital T, lowercase t, and it says both of the parents are like that, so parent 2 is the same, capital G, lowercase g, capital T, lowercase t. The handy tips for solving the problems is identify the parent genotype. Check, check, we did that. Identify all possible gametes from each parent. So going back as to part 2, this was our problem, the possible gametes from parents. So for parent 1, the gametes, the capital G can combine and assort with capital T. The capital T can sort with lowercase t, as well as lowercase g can sort out with capital T, and lowercase g can sort out with lowercase t. So these are the 4 possible gametes. Now, identify possible gametes. Check. Determine the dimension of your Punnett square by counting the possible gametes. Now, this is what I did not say in the previous practice, but please check it. If we have a heterozygous, for example, for the both two characters, now how do we figure out? This is the capital, the G's can sort out in 2 different ways, either capital or lowercase g, and the T's can sort out capital T, lowercase t. Now, because they are independent from each other, so we time them, so the all possibilities will be 4 different gametes. Please apply this one, this idea for the part 2. Check this one, A and A, it can sort out in only one way, and the B's can sort out in 2 ways. 1 times 2 is 2, so we have 2 types of gametes in here. So please check back to find out that. So moving forward, these are the possible gametes we will put at the top of the big Punnett square. This is one parent, and the other parent will be on the side. And please pull those gametes together. What I would do, just sort out the G's first and the T's, and use the capital letter first for that type of a letter. So this will be capital G, capital T, lowercase t. I might just pause it and fill. So here is the filled out Punnett square. Let's see, just use highlights for green ones. This is going to be green, 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 green. These are all going to be green in colors. Okay, and so this is going to be green. If it's going to be yellow color, these are going to be and height wise, blue will be tall. These are going to be the tall plants. Okay, and what is the phenotypic ratio of the offsprings? So we have the green, tall, green, short, yellow, tall, and yellow, short. All together we have 16, so we have green, tall, 1, 2, 3, 4, 5, 6, 7, 8, 9. Oops. This is not, so 1, 2, 3, 4, 5, 6, 7, 8, 9 of them is going to be green tall. Green short, so green color, but not colored the T. So this is 1, 2, and 3. Yellow tall, so I'm looking for a yellow color. Oops, yellow. 1, 2, 3. And the yellow short is only one of them. So the phenotypic ratio is going to be 9, 3, 3, and 1. What is the frequency of yellow-seeded offspring? Yellow-seeded, short and yellow. This is the short and this is the yellow, so it's going to be 1 over 16. 16, and only one of them will be that. What is the frequency of the tall green-seeded offspring? Tall, capital T's, or homozygous or heterozygous, dominant for that. And the green is colored green, either contains 1 or 2 capital G's. It's going to be 9/16. Now, alternatively, we could have considered the two traits separately. Let's check if we are using the small Punnett squares, one for the seed color, oops, and for height. Both of the parents are heterozygous for that seed color and for height. Please complete the Punnett square. And it's asking us, what is the short yellow? The short and yellow, that's the yellow color. Oops. Come on, yellow. This is going to be yellow and short. I don't know what color I used that. Anyway, this is going to be short. So in here, 1/4 is going to be yellow times, because they are independent from each other, and the short is 1/4. And 1/4 times 1/4 is 1/16. That's the same as we got over there. Sorry. Now, the other one, the tall and the green seeded. Green seeded 1, 2, 3, and 3/4. Right? It's going to be green seeded. And tall is 1, 2, 3. It's a 3/4 again, and independent from each other because they are on two traits separately. They are independently assorting. We time them together. 3 times 3 is 9, and 4 times 4 is 16. That's the same number as we got here at the top. Now, in pea plants, the green color allele is dominant over yellow allele for seed color, and tall is dominant for the allele in a plant height. So that's the same prompt what we had before. One homozygous recessive, so the parent one is homozygous, lowercase g, lowercase g, lowercase t, lowercase t, is crossed with parent 2 that is, is short and heterozygous for color, capital G, lowercase g, and short because it has 2 lowercase t's. Create a Punnett square to determine all potential offspring genotypes. So in this case, the gamete possibility is only lowercase t, lowercase g, lowercase t. For this, gamete possibility is capital G, lowercase t, or lowercase g, lowercase t. So how we are going to do the Punnett square? Just kind of, oops, even smaller. Even smaller. 1, 2. So parent 1 will be GT in here, and capital G, lowercase t, lowercase g, lowercase t. It can be this, or it can be that. So the gametes are those, and the Punnett square should be 1/2. Offspring genotypes can be green genotypes or phenotypes. They can be green seeds and short plant, or yellow seeds and short plants. These are the only possibilities, and 50% is the frequency or the chance. What is the probability of each of the following sets of parents producing the given genotypes in their offsprings? So for this part, the probability of each of the following sets of parents produce the given genotype of their offsprings. Get back visual, just how it looked like when we crossed to heterozygous. That was half of them was heterozygous. The genotype frequency was 1 to 2 to 1, so it's going to be half the probability. When we crossed heterozygous with homozygous recessive, in this case half of them was heterozygous. If we cross this separately, we check for the A's, 1/4 was homozygous. 1/4 was homozygous dominant, and when we do separately, like we did in here, the cross for B, in this case, half of them was homozygous dominant, and we time them together because they are independently assorting, and what we will go is 1/8. The next question is interesting because we are looking for lowercase a, lowercase a. It's not going to happen because parent 2 cannot contribute a lowercase a for the offspring, so the probability is 0. And here, heterozygous with heterozygous to produce a heterozygous for both traits, it's going to be in here half is the probability if we independently assort them times half, oops, for the other cross 2, and half times, oh no, half times half is 1/4. Okay, so number 11, if an offspring has the genotype of AA, what possible combinations of parental genotypes could have produced this offspring? So the parent must contribute an A and the lower k for that offspring. So the first combination, so this will be the outcome, A slash and A something. So it can be AA times AA. It can be AA, oops, times AA. It can be capital A, lowercase a times lowercase a. I'll put the capital in front of it. And we can have heterozygous one parent crosses with homozygous recessive. These are the 4 different parental crosses. Now, in corn, the allele, so number 12. In corn, the allele for the tall plants is dominant for the dwarf plant, and the allele for the colored kernel is dominant, colored, dominant over white, in a particular cross of a corn plant. The probability of offspring being tall is half. When was the half happened? When one parent was heterozygous and the other parent was homozygous recessive. And the probability of the kernel being colored is 3/4. That was the case when we had two heterozygotes cross for the color. So heterozygotes and heterozygotes for the color. This could be the parental genotype for that cross. Number 13 is in a cross between a parent with genotype and a second parent with genotype, what proportion of the progeny will be the same phenotype as the first parent? Let's do it together. I would like to write it down again, how the parent looks like, parent 1. Parent 2, homozygous recessive, heterozygous, and homozygous recessive. The question is, the offspring needs to be like the first parent. Looking like that. So what is the probability for the A when we cross this to have a dominant, shows a dominant phenotype? It's 3/4. For having a dominant phenotype for B is 1/2. Having a recessive phenotype for C is 1/2. To have a dominant phenotype for D is 3/4. And for recessive phenotype for E is 1, because they are independent from each other. We time them all together, and if we time them all together, 3 times 3 is 9, and 4 times 2 is 8, 16 times 4 is 64. The chance is 9/64 for that the progeny will be the same phenotype, not genotype, but phenotype as the first parent. And that's the end of part 3.